Hello, my name is Bruna Bastos, and I am an undergraduate student at the University of Brasília. I'm here to present critical appraisal tools used in systematic reviews of in vitro cell culture studies, a systematic review. Systematic reviews are on the top of the evidence pyramid, and preclinical studies are on the base. The in vitro techniques are used for organ analysis, tissues, and cells. So, usually these techniques are used in pharmacological and biological fields, and it is the precursor for clinical investigation. In the other way, systematic reviews are performed to answer a specific research question through the selection and assessment of primary studies as observational, laboratory, and clinical studies. Applying appraisal tools to analyze the methodological quality of included studies is an essential step in conducting a systematic review. Such applications are recommended by guidelines as PRISMA 2020, and this recommendation is mostly because the assessment provides an outline of the quality and risk of bias of the research and increases the potential for informing clinical recommendations. There are a range of tools for clinical studies. However, assessment tools for some types of studies, such as cell culture, are still lacking. So, the objective of this methodological systematic review was to identify, collect, and analyze systematic reviews involving cell culture studies to highlight the appraisal tools currently utilized. The electronic search was performed in February 22 on Embase, PubMed, Scopus, and Web of Science. The search ranged from 09 to 22. About the study selection, we included the systematic reviews of in vitro cell culture studies that used the critical appraisal tools for methodological quality or risk of bias assessment. The selection process was performed by two independent reviews in two phases. In each study, we collected the basic information. The methodology quality assessment was carried out by two authors independently using Joanna Briggs Institute critical appraisal checklist for systematic reviews and research synthesis. Based on eligibility criteria, we included 82 studies in this review. Of the 358 reports excluded, 72% studies were excluded for not present critical quality appraisal of primary studies, indicating low adhesion to an essential step of a systematic review. Over time, there was an increase in the publication number of systematic reviews of cell culture studies that assess the methodological quality of primary studies. There was just one publication in 2011 and 40 in 2021. From the 82 studies included, 49 adopted the pre-structured tools, 23 used modified tools, and 10 self-developed tools. The two most frequently used were Circle, Systematic Review Center for Laboratory Animal Experimentation. Pre-structured tools lack important questions when analyzing cell culture studies, although authors may have thought that self-developed tools would fill the gap in methodological quality assessment of cell culture studies. 63 studies achieve high methodological quality, while 19 reached low. And based on tools used in the included studies, we highlight some important criteria and questions that can be used in the future in evaluations of in vitro studies. For example, regarding the scientific background, it's important to report the rationale and objectives of the study. Also, the sample, the conditions, and all procedures needed to have a clear description. Some studies need ethical approval, and it's important to have a control or standard reference. Investigators can be blinded, and samples can be randomized. It's also important to make analysis between groups, present the justification of method analysis, and evaluation by independent observers. About the data collected, confounders need to be adjusted, the outcomes need to be accessible, the results have a critical discussion, and the conflicts of interest declared. In conclusion, our study identified and analyzed currently used the critical appraisal tools for methodological quality and risk of bias assessment in systematic reviews of in vitro studies with cell culture. Further, these results may serve as basis for the development of a validated critical appraisal tool for in vitro cell culture studies, contributing to evidence-based knowledge generation. Thank you so much for your attention.